Hello! Ever wondered why we have trillions of cells inside our bodies? That's because the cells have their own process of dividing to reproduce more cells. In this video, I will be talking about the process of mitosis. First off, what is mitosis? Well, mitosis is a type of cell division. It is a process where a single cell divides into two identical daughter cells. Mitosis is divided into 5 phases: interphase, prophase, metaphase, anaphase, and telophase. The interphase. Interphase has 3 phases within it: the G1 phase, the S phase, and the G2 phase. The G2 phase, as you can see in the picture, the cell has 2 centrosomes, and each has 2 centrioles. And the string-like thing here inside the nucleus is the DNA that has already been copied. At this stage, the DNA is surrounded by an intact nuclear membrane, and the nucleolus, here in the picture, is present in the nucleus. Here, in this phase, the cell grows more and organelles and proteins develop in preparation for cell division. In the S phase, the DNA is being replicated, and each of the 46 chromosomes is replicated by the cell. And lastly, in the G1 phase, the daughter cells enter. There are 2 daughter cells here. The cell increases in size, and cellular contents are duplicated. After the interphase is the prophase. In this picture, the mitotic spindle starts to form. This one here. The chromosomes start to condense into X-shaped structures that can easily be seen under a microscope. And the nucleolus that was once here disappears. Next to prophase is the metaphase. Chromosomes line up at the metaphase plate, an invisible line in the middle, under tension from the mitotic spindle. The 2 sister chromatids of each chromosomes, here in the middle, are captured by the microtubules, this one, the string, from opposite spindle poles. Anaphase. The sister chromatids separate from one another, there, and are pulled towards the opposite poles of the cell. The microtubules that are not attached to the chromosomes, here, push the 2 poles of the spindle apart, while the kinetochore microtubules are the ones who are pulling the chromosomes towards the poles. The last phase is the telophase. The spindle disappears, and the nuclear membrane reforms around each of the chromosomes, and a nucleolus reappears in each nucleus. The chromosomes also start to decondense. It's no longer in the X shape. Cytokinesis happens to start either in anaphase or telophase and finishes after telophase. Cytokinesis in animal cell. Here, an actin ring around the middle of the cell pinches inward, creating an indentation called the cleavage furrow. The plant cells, on the other hand, can't be divided like the animal cells because they have a cell wall, and they are too stiff. Instead, a structure called the cell plate forms down the middle of the cell, splitting it in 2 daughter cells, separated by a new wall. When the cytokinesis finishes, we end up with 2 new daughter cells, each with a complete set of chromosomes identical to those of the mother cell. The daughter cells can now begin their own cellular lives, and depending on what they decide to be when they grow up, they may undergo mitosis themselves, repeating the cycle. So that's all for today. Thank you for watching.